Welcome to the 25th lecture on environmental biotechnology. This particular lecture will be on methods in microbial ecology with reference to environmental biotechnology. In the particular lecture, we would be discussing about the microbial ecology related methods and the major developments that occurred in the past 50 years or so. We will discuss in detail about the uncultivability of organisms, and we will begin with the great plate count anomaly and different categories of uncultivable or cultivable organisms which are present in any environment will be discussed. So, to begin with, the long term goal of microbial ecology is to gain a better understanding of the ecology of important microorganisms in environmental samples. And as we realize that for any successful environmental biotechnology process development, understanding the microorganisms with respect to their ecosystems is a critical component. Microbial ecosystems are a sum total of all the organisms and different type of physical and chemical factors present in a particular environment. So, it is a basically dynamic complex of not only microorganisms but also the plants, animals, their products and the abiotic surroundings. An ecosystem therefore contains different habitats. A particular habitat is a part of the ecosystem, typically refers to a zone in which a particular species or populations naturally lives and grows. So, with reference to methods, those are applied to understand, characterize and make use of these microorganisms present in any environment towards sustainable process development. This basic information about the ecosystem concept and habitat is very important. So, here you can see a couple of interesting ecosystems or the zones which are very relevant for environmental biotechnology related process. To begin with the agricultural paddy system, a naturally petroleum oil spilled environment within a forest area, an acid mine drainage producing system within a mine dump site where highly acidic and sulfate rich mine drainage is flowing through from the acid mine waste dumps, and below here is another very interesting ecosystem which is basically showing a mine tailing dam rather, and all these ecosystems are subjected to biotechnological interventions for either crop productivity or bioremediation or sustainable recovery of metals and minerals or mine activities, etc. So, within all these different kinds of environments or ecosystems, what we observe that the types of microbial activities and the rates of the microbial activities are of high significance, because those are the processes which are controlling the biotechnological or going to control the biotechnology related process. So, now when we plan for environmental microbial ecology methods, the methods should be selected in such a way so that they are able to identify the different type of microbes and also help us to elucidate the rates of individual microbial activities. So, the type of the microbial activities could be delineated as a function of the species present, their population sizes and the physiological state of the microorganisms. To elaborate this particular point briefly, so in any environment, whatever microbial activities are expected, that is basically an outcome of the function of the individual species present. So just few minutes ago we were looking at a petroleum oil spill site within a forest area. So within that highly petroleum oil contaminated environment, what type of microbial activities are expected, or whatever type of microbial activities are predominant, can be attributed to different type of functions which are carried out by individual species member. So in a nutshell, the community function or the ecosystem function, whatever is obtained for any kind of environment, is basically an outcome of the individual functions of the species and also their interaction with the abiotic surroundings. Now the individual species function is of course a very important component, but at the same time the size of those individual populations are very important. For example, in case of a petroleum oil contaminated site, there may be some species who are capable of degrading the long chain alkane or petroleum more complex hydrocarbon molecules, but those species, let us assume there are certain Burkholderia or Pseudomonas species who are capable of degrading the very long chain alkanes and complex aromatic compounds. But the size of those, population size of those Burkholderia and Pseudomonas are of a great concern, because there may be few Pseudomonas or Burkholderia who are capable of degrading those long alkane chains or the complex aromatic compounds, or there could be a high population size of those organisms. So if the population size of those Burkholderia and Pseudomonas are higher, we can expect that within that particular petroleum oil contaminated environment, an enhanced activity towards the degradation of the long chain alkanes or the complex aromatic compounds will be achieved. However, if the population sizes of those Burkholderia and Pseudomonas species are low, then it may be found that although such organisms are present, because of their abundance which is very low, the actual degradation of the long chain alkanes and polyaromatic compounds are not up to the mark. The third point is also very important, which is the physiological state of the microorganisms. Again, if we take the same example that in a petroleum oil contaminated environment, we may have the Burkholderia species who are very much capable of degrading the long chain polymeric or long chain alkane compounds, but due to some restrictions or some constraints, the physiological state of the those Burkholderia species who are capable of degrading the oil molecules or the alkane chains are not appropriate. So they are not in a favorable physiological state so that they can express the genes or the enzymes which might facilitate the degradation of the compounds. So therefore, the activities that we are expecting in any kind of environment must be understood with respect to these three important parameters. The second important aspect is the rate of the activities. So there may be, in this case that we are discussing, the petroleum contaminated site with Burkholderia species capable of degrading the long chain alkane molecules. They are of significant population size and their physiological states are also well maintained. However, if they are not provided with the required nutrients, for example the nitrogen or phosphorus or the phosphate molecules, or there are other growth conditions, for example the temperature or the pH are not optimum or not within the favorable regime for those specific group of microorganisms, then even if the specific microorganism is present, their population size is significant and their physiological state is apparently perfect, but they are not going to function adequately because they need other nutrients and the growth conditions which are not available. So therefore, when we develop any particular method, we should be careful that our method should be able to delineate or decipher both these types of the activities as well as the rate of the activities. Now, the impact of the microbial activities within an ecosystem depends on several other factors that we might discuss in or might have discussed already in some other lectures. Now, coming to the original idea of talking about the methods of microbial ecology, which are very relevant for understanding any ecosystem or environment where biotechnological processes are to be implemented or developed. So, there are two components within these methods. So, the methods could be targeted towards understanding the biodiversity, that is the kind of organisms which are present over there. So, in a simple word we can say that this is basically the method which will, or set of methods which will allow us to understand the biodiversity of an environment, of a particular system. Maybe an wastewater, maybe a contaminated lake, maybe a underground system where some carbon dioxide is going to be sequestered, or in any kind of other environments. So, biodiversity will give us a general broad picture about what are the organisms present. But with respect to the role of, or delineating the role or importance of the significance of the microbes which are present there, the identification of those microorganisms, all microorganisms present in a particular environment, and quantification of those organisms. So, identification and quantification are those terms which are very often used and are very important with respect to microbial ecology and environmental biotechnology. So, if we understand that in a given environment or a subset of the environment or within a habitat of an environment, we have 100 different species, then each of these 100 different species need to be identified. Identified means their taxonomic identification must be obtained, and quantification means that each of those 100 organisms or whatever may be the number, once we identify the organisms, who are these organisms, we must be able to tell what is the quantitative abundance of each of these species. There may be Pseudomonas, Bacillus, Microbacterium, Burkholderia and Rhodococcus and many other bacterial species, archaeal species, fungal species, everything is there. So, we should be able to tell how much of the Pseudomonas and how much of Burkholderia and how many are the methanogenic archaea, individual species are there. So, this is not so simple, because microbes are so tiny organisms that under the microscope, even if you see them, it would be hard to actually precisely identify. You can count the total number of cells, but it is hard to count numbers of a specific type of cells or specific organism or specific taxonomic group. So, microbial ecology methods have been developed which help us to not only to identify the organisms but to quantify each of the type of the organism. Even if there are many more organisms, like exceeding few hundreds different organisms, it is possible to quantify each of the species member within any environmental sample. So, this is the part on the biodiversity part. On the other hand, with respect to microbial activity, that is another important aspect of microbial ecology methods. With respect to microbial activity, as I have mentioned earlier, that the activity, type of activity, rate of activity, there are many other parameters within this microbial activity. So, with respect to microbial activity, the microbial ecology methods try to measure the microbial metabolic activities, that means the functions carried out in their habitat. So, if we consider that in a particular acid mine drainage environment, there are certain sulfate reducing bacteria who could be useful for precipitating the sulfate, because once the sulfate is reduced to sulfide, the sulfide precipitates. So, if we want to assess or monitor the activity of those sulfate reducing bacteria, so we need to actually assess the enzymes which are involved in sulfate reduction or the other properties which actually will connect us to the metabolic activities, metabolic processes of sulfate reducing bacteria within that mine environment. Possibly, if you are able to do that, with suitable methods, we will be able to utilize such microbial activity for the remediation or reclamation of a contaminated mine site. Now, in this case, with respect to the methods of microbial ecology, there are 4 fundamental questions of microbial ecology that we have already discussed earlier. These 4 questions are to be answered. So, our microbial ecology methods should be able to answer who is here. That means, who are the organisms who are present here? What can they do? This is related to the question that we earlier framed as what is the potential of this microorganism that they can possibly do, and then what are they doing. So, these second and third questions are interrelated, but they address or they provide answers which help us to understand the role of the microbes at the different level. What they can do is the kind of a potential that provided something happen or something not happen, they will be doing this. But what they are doing is actually a real time phenomenon, that actually what they are doing when we collected the sample. And lastly, how do they interact and work as a system? So, there may be, as I said, 100 or 1000 or few thousand species member in a particular environment, and many of them are capable of or doing multiple functions. So, these functions and their presence of the organisms, they must be interconnected to each other. Now, when they are interconnected to each other, they must be working as a system. So, how we understand that system so that we can exploit them to the best of our possibility. So, the first question, that is the who is here or what are the organisms present there, would lead to the deciphering the particular point which is called community composition. So, when we develop a method or use a method, microbial ecology method with respect to a particular sample to answer the question that who are there or who is here, we basically get the answer about the microbial community composition. In subsequent lectures, I might use the term microbial community composition or microbial community structure. So, that refers to what are the organisms present there. So, it includes both the identity of the organisms, as I mentioned earlier, and also the quantitative abundance, so relative abundance of the species or the taxonomic groups who are present there. The second is the phenotypic potential, which is connected to what they can do. So, phenotypic potential can be assessed by a number of methods, enzymatic assay or providing different substrates under laboratory conditions. So, phenotypic potential is basically a kind of assessment that these organisms could potentially be able to do this. But we should keep in mind that these assessments are done under laboratory condition. So, under laboratory condition, a particular microbe may respond to substrate differently compared to a real situation, because in a real environment there could be lots of other parameters, because that is not a controlled laboratory environment, and also there could be innumerable number of other species, other organisms and other physical and chemical factors which will be interacting with those species. So, the real activity of the metabolic activity or metabolic function of the organism might be different. So, we need to actually address that what are they doing. So, when we address this question that what are they doing, we basically try to refer community function. So, it is not an individual's role, it is the community role, because even if there is a very important or a major bacterial species or archaeal species or a fungal member, in a real environment, this particular fungal species or bacterial species would be interacting with many other species and also in presence of many other organisms. So, when we try to answer this question using an appropriate method, that what are they doing, we basically derive the community function. And finally, the interaction among the organisms, which refers to the systems level understanding. So, the questions that we ask, that which bacteria and archaea, the microorganisms in this case, and including the fungi and protozoa wherever it is applicable, like a wastewater treatment, perhaps we have to even include virus also, so present in the sample. So, if you are dealing with an activated sludge or a wastewater treatment, or we are looking into a drinking water system, we might have to consider all possible type of microorganisms who are present there. So, our method should be able to identify them and also find out the quantitative abundance. So, how many types? So, when we say how many different types are there, so possibly that answer is obtained when we say that the identification of the species are already done. So, when you have identification of the species members are done, then naturally the taxonomic types are obtained. But there could be another categorization which is called a physiological characterization. So, earlier we have studied the different carbon and energy source requirement. So, a broad categorization of the physiology of the microorganisms are with us. Either they may be heterotroph or organotroph, or they may be autotroph, they may be chemoautotroph, they may be photoautotroph, they may be lithotroph. So, those are the different kinds of physiological categories or types of organisms present there. The third question, which microorganisms are active and growing? Because often, whether it is a drinking water system, whether it is a wastewater treatment system, whether it is a contaminated lake or river or any kind of other environment where we are trying to do environmental biotechnology process, we collect sample, bring the sample in the laboratory and test them. So, in this case the testing is with respect to different organisms who are present. So, we may identify the different organisms and we can also identify their physiological types by appropriate methods that we will discuss. But these are all done under laboratory condition. Actually, in the real wastewater or real drinking water, which are the microorganisms or what are the microorganisms which are actually active and growing? Because I may bring the water from the field, and when I test I may find out organism A is more active and growing very fast, but actually it is other way around. It may be organism C or D is actually more active in a real situation, because as I already mentioned and you perhaps understood that the real environment are different from the laboratory environment. So, whenever we develop method, we implement method, we must be very careful about this, that we have laboratory constraints. So, when we incubate a cell or a microbial culture under 30 degree or 37 degree and use a particular medium with a definite carbon and nitrogen source, a pH of the medium is also very much controlled. These are not always replicating the exact scenarios or exact situation in the real environment. So, environmental biotechnology processes must rely on real situation. So, methods must be selected, methods must be developed which will address the issue like which microorganisms are actually active and growing. The next question, what is the ecology of microorganism in the context of their environment? It is a very difficult or very complex question, because you might be able to identify 10 or 20 or 100 or maybe 500 different organisms. You may be also able to physiologically categorize them, that there are 10 different physiological groups of organisms are there. You may also apply certain methods, we will talk about those methods, that these out of the 100 organisms present there, 100 bacterial species present there, only 20 are active and growing in the real environment. Yes, we can do that. But what is the ecology of the microorganism in the context of the environment? In order to understand that, we might need to understand the environment itself, because it is more complex than we perceive, and then we need to actually implement such methods which will help us to understand the ecological significance of organisms, because in most of the environmental biotechnology processes are multi-organismic. That means there are more than one microbes involved in environmental biotechnology process. So, when we have more than one microorganisms, for example, conversion of waste to alcohol or maybe methane or other useful compounds which are industrially or energetically very useful products. So, these, as we already discussed, these are all by network of organisms, network of metabolic processes, both the primary fermenter, secondary fermenter, sulfate reducers, methanogens, many different types of organisms are there. So, we need to understand the complex process of microbial interaction within the environment itself. And then finally, answering this last question, that how can we apply this knowledge in terms of bioremediation, in terms of fermentation, in terms of other industrial application, that is also a very important aspect for the development or implementation of the method. Now, with respect to microbial ecology methods, there are three main approaches. First is the in situ or the field observation. So field observations are, we can go to a contaminated site, or we can go to a lake or a river or a site where some kind of environmental biotechnology process is going to take place or is possibly working there naturally. So we need to observe considering the in situ conditions with minimal disturbance to the studied processes and communities. So we need to implement certain strategies that will provide us information which are in situ. The second is the laboratory and field experiments with deliberate modification of the natural object aimed at revealing of unknown functional relationship. So in this case, we bring the sample into the lab and then expose the sample or incubate the sample under different conditions. That is the modification. So deliberate modification means we try to provide different carbon source, different nitrogen source, different pH, different redox condition. In earlier lectures we have seen how the redox condition affects the electron acceptor domains. So we can provide actually different electron acceptors by creating different redox conditions, and then see how the system, the soil and its or the contaminated waste or the environmental sample is reacting to this changing redox regime and with respect to the electron acceptors provided, and from that the unknown functional relationship can be obtained. The third approach is the mathematical and conceptual modeling based approach, which aimed at generation of new theoretical knowledge based on the interaction between the species, the functional properties, the enzymes, etc., testing different hypotheses and comparison of the theoretical concepts. And actually this last aspect, that is the methods which will help us to understand the develop conceptual model, is considered as a source spot. Relatively weak development of theoretical concepts and inadequate laboratory surrogates are provided. Now, how to study the microbial diversity within a particular environment? So we will proceed into that how microbial communities are actually deciphered with respect to answering the taxonomy, the diversity, the quantitative abundance and their activities, particularly 4 questions that we have framed. But there are certain general limitations with this respect. So one of the most important limitation is that the lack of taxonomic knowledge, how to identify it, and that is a kind of methodological limitation, because microbes are so tiny, they are so small that by looking at the microscope, through a microscope you cannot identify exactly what is this organism, whether this is a streptococcus, staphylococcus, what type of organism it is. It is not always so simple. Pseudomonas and bacillus, sometimes they appear almost same morphologically under the microscope. So it will be hard to identify them. So there should be method which will help us to identify the organisms, and if we consider that the diversity of the microorganisms are very, very high, a gram of soil might have several thousand different species. So if we have several thousand different species within a gram of soil sample or a milliliter of water, 1 milliliter of water having 5,000 different species. So it is very challenging to identify them, 5,000 different species. Then there is a point of spatial heterogeneity. It is a kind of a considered as an innate heterogeneity and spatial distribution of microbes, particularly in soil, sediments, etc. These environments are highly heterogeneous. In each millimeter or even less than that, there are small microhabitats, and these microhabitats are allowing growth and proliferation of specific group of organisms, including bacteria, archaea, fungi, even protozoa also. So this is, and these microhabitats vary over a small to broad range of length and space and time also. So spatially and temporally this heterogeneity varies. And also there is a nested level of organization that could be dependent on different physicochemical properties. A small microhabitat having a set of organisms proliferating when the pH is low, but the same microenvironment will have a different microorganisms, or the activity of the microorganisms will change as soon as the pH is altered. The product of this small microenvironment, the kind of metabolites produced, the kind of changes they bring into the iron and sulfur and other redox active elements, these also affect the activities of other organisms. So therefore, the entire organization is highly nested. One microhabitat or microzone is connected to another one. Because of the microbial activities, one produce certain things and these products are utilized by the other organisms. So after considering all these things, we have realized that the prokaryotic microbial diversity is one of the largest knowledge gaps in the biological sciences. So on the one hand, we have a large number of questions in front of us. We have understood the complexity of the environments, be it a soil system, sediment system, river system. In most of these systems, we have a very much heterogeneity, and this heterogeneity is both spatial and temporal and also nested in nature. And so overall, the deciphering the microbial community with respect to the diversity and its activity, which are very important for developing and implementing any environmental biotechnology process, is identified to be largely unexplored and unexploited. Now, why it is so? So if we look at the methods in microbial ecosystem studies and the major events of developments, and we begin with the very well known phenomenon of Koch postulates by Robert Koch in 1884, who put forward the concept of pure culture. Organism which is unadulterated, is not mixed with any other organisms. This is a pure bacteria, archaea or fungal culture. Only one particular strain or species is present there. So that was long time ago, 1884, the importance of pure culture was established. But the pure culture was not enough, because in real environment there will be thousands of different species together. So very soon Winogretsky deciphered the concept of chemoautotrophy and how microorganisms behave in a more complex way than possibly we perceived. And then subsequently next, I will say 100 years or so, there are many reports, many studies with respect to environmental biotechnology and environmental processes. Most of these studies were targeted towards isolation of cultivable bacteria. Cultivable bacteria meaning the bacterial strains which are capable of growth under laboratory conditions. So you can grow them under laboratory condition, that is on a agar plate, on a broth medium, you can grow them, you can see the tiny colony growing on the agar plate from different contaminated sites and studying their degradation and other metabolic processes. So a large number of studies in that particular period, last 100 years or maybe little less than that. So huge number of studies have been done. So many bacterial and archaeal species were isolated from different environmentally relevant sites and characterized. While we were isolating, or the scientists working on the environmental biotechnology or environmental microbiology or environmental engineering were isolating organisms, identifying them in order to understand the quality of a drinking water or the performance of an activated sludge treatment or a wastewater treatment process, there were some parallel developments happening. So one of the major development that was happening in the corner, it was the ribosomal RNA based phylogeny. So we will talk about this phylogenetic analysis little later, but these are the small stretches of DNA within the genome or the chromosome of the microbes which code for the ribosomal RNA, and ribosomal RNAs are integral part of ribosomes, and ribosomes, you know, they are the machineries useful for the translation. So without ribosomes, translation will not happen, and the ribosomes are made up of ribosomal RNA and ribosomal proteins. So this ribosomal RNA along with ribosomal protein sequences, both these sequences were used to understand the molecular evolution of microorganisms during the time of 1997 and all. So at the same time or subsequent to that, or even before that, we had a very well defined concept of 16S ribosomal RNA. That is, one of the ribosomal RNA molecules, that is a 16S ribosomal RNA molecules, was identified as a phylogenetic marker, that this particular gene can be used. So, this 16S ribosomal RNA is a part of ribosome, but it is encoded by a gene in the chromosome. So, that particular gene is referred as 16S ribosomal RNA gene. So, that particular gene can be useful for identifying an organism. So, earlier I was talking about identifying a microorganism by looking at the microscope. So, looking through microscope, it is very difficult because microbes are all or almost all cases they are either bacilli or cocci or streptococci or something like that. So, it is very difficult to discriminate the different types of microorganisms, particularly when we consider that there are more than few thousand species present there. So, 16S ribosomal RNA was found to be very useful in identifying the microorganisms. So, we will talk and learn about this little later. However, during this time 1985 or so, there was a very interesting discovery, I will say, or an interesting report came which refers to great plate count anomaly by Staley and Konopka. Now, what is this great plate count anomaly that we are going to talk today? But this great plate count anomaly refers to the anomaly with respect to number of microbes that we count through a simple agar plate method, where we spread a sample on an agar plate and try to count how many cells are there or colonies are there. At the same time, when we see this sample under the microscope and try to see how many cells are there. So, these two numbers never match. The number that we obtained from direct microscopic count is found to be always higher, at least 100 to 1000 times higher than the number that we get as colony count from the same sample. That is called the great plate count anomaly. So, from the great plate count anomaly, the concept of uncultivability or unculturability of natural microorganisms evolved. So, that means we will discuss this, but to have a very preliminary idea that microscopic count is providing more number of cells, but the agar plate based method or the colony count method is not providing so many cells, and the number of cells are at least 2 orders of magnitude less. Like if one cell is coming in a CFU, colony forming unit, microscope will provide you at least 100 cells. So, what is going wrong? So, finally, the scientists they found that it is the uncultivability of the natural microorganisms. So, when we look at the sample with microscope, so there are 100 cells. Most of these cells, almost 99% of these cells are not capable of growth under the laboratory condition. So, they are uncultivable, and this is a property, or this is a kind of a manifestation of a property which is called uncultivability. So, many organisms are uncultivable, they do not grow under laboratory condition. So, as soon as the 16S ribosomal RNA based methods appear and they are streamlined because of the sequencing development in PCR and sequencing techniques. So, microbial community analysis, structure and function both were streamlined, initially using the 16S ribosomal RNA gene, followed by the different functional genes that we will discuss in detail in the later lectures. Then the metagenomics, the concept of metagenome came, where all the organism's genome can be studied together. And finally, it is not only the metagenome, that is the collection of all the genomes present or the organisms present in a sample, but also the total protein that is called the proteome, or the total RNA that is the transcriptome or the metatranscriptome, or even the total pool of metabolites produced by a particular community that is called metabolome, and even the reconstruction of the genomes of the uncultivable organisms were possible, and probing the role of individual species and populations or guilds were possible in today's time. So, there is a kind of a huge, or we call a paradigm shift in microbial ecology or environmental biotechnology methods, starting from the pure culture bacteria isolating or looking for few known organisms in your sample, to a whole lot of omics approaches and a kind of all-inclusive studies where every organisms, every possible processes can be identified and studied. Now, if we look at this uncultivability, one of the major point is the cause is the failure of microbiologists to describe natural diversity. So, because the morphology, as I mentioned, it is too simple to serve as the basis for classification system, and until very recently a successful microbiologist was very much determined by the ability to cultivate microorganisms, followed by characterization of multiple physiological and biochemical traits. So, it became a kind of a routine practice that in order to decipher the microbial diversity or microbial property of a particular environmental sample, any microbial ecologist or environmental biotechnologist used to rely on isolation of few cultivable bacteria or archaea or fungi, followed by their characterization and their further analysis. Now, it is understood that any approach to identify the specific microbial populations without cultivation, because now we have realized that 99% of the cells or the species present in an environment are not cultivable. So, any approach that will identify the specific microbial populations without cultivation directly from their natural environment is therefore considered to be a revolutionary process. It would actually change, and it has actually changed, the character of environmental microbiology, and it has tried to close the methodological gap which still exists in comparison to other branches of biology, particularly the botany and zoology. Now, this revolution of microbiological thoughts over the past 30 years, if we see, one of the best examples is the great plate count anomaly, as I mentioned earlier, that is the discrepancy between the size of the populations estimated by dilution plating and the microscopy. So, in one hand, the samples are taken, diluted and seen under microscope, and the direct microscopic counts are obtained. So, we observed that the direct microscopic counts exceed the viable cell counts by several order of magnitude, and the majority of the microscopically visualized cells are viable because we have methods to identify whether those cells are viable. We find that they are actually viable, but they do not form visible colonies on the plate. So, for example, in this simple example, we can see that preparation of a soil dilution in a sterile buffer. If we take a soil, 1 gram of soil and dilute it serially up to 10,000 times, and then plate this, dilute maybe 100 microliter if we sprayed onto this, as well as 100 microliter or a subset of that, we can see under the microscope. We will see that if under the microscope we are able to count maybe 100 cells or 1000 cells, in agar plate only one cell is going to appear. So, what I was referring earlier, that 1 out of 100 cells will actually grow on agar plate or give a viable count. So, in a subsequent study, Aman et al. Reviewed and found that in different environment, including sea water, fresh water, lake, unpolluted water, sediment, soil, even activated sludge, the cultivability varies, but it almost in all cases remain close to 1% or less than that, except the activated sludge, because in activated sludge, you know that we use a particular inoculum or a particular set of organisms who are only growing there continuously, and they are cultured under that condition only. So, the cultivability of the organisms are little higher in activated sludge, but in rest others it is close to 1 or even less than 1. So, two different types of cells are found to contribute to these silent but active organisms, because 1 out of 100 is able to grow as a viable cell. So, rest 99 cells, they are viable, but they are not growing. The picture on my right side shows basically the epifluorescent microscopy of a soil microorganism stained with DAPI, and you can see the numerous cells which are present in the sample. Anytime anybody can take a sample from a soil or a water and stain with DAPI or similar fluorescent dyes, DNA staining dye, and see under microscope, and you will be able to, or she will be able to count how many cells are there. But if you take a same sample and dilute and plate on an agar plate, you won't be getting so many counts. The cultivability will be definitely within this range. So, it is considered that two types of organisms must be contributing. One is a known species for which the applied cultivation conditions are not just suitable. So, whatever medium or media we have used, or the pH or temperature etc. we have provided, it is not suitable. The other could be that these organisms are actually unknown species that have never been cultivated or cultured before, and the lack of suitable methods. Now, the major points to note with this respect, or in this respect, that 0.01 to 1% of the total bacteria in particular are cultivable. 70% of total bacterial phyla, they do not have any cultivable representatives, and our understanding of microbial diversity is not represented by the cultured fraction of the diversity. So, the cultured microorganisms represent only a small fraction of the natural microbial communities, and hence the microbial diversity in terms of species richness and species abundance is grossly underestimated. Now, with respect to inability to culture these environmental microbes and hence their identification, characterization, environmental role and significance, it is possible that this 1% of cultivable organisms, they are representative of the entire population, and that the other 99% are simply in a physiological state that eludes our ability to culture them. But at the same time, it could also be possible that the 99% of the cells are phenotypically and genetically different from the 1%, and only the minority of the population is represented. In most of the cases, in the later studies using different environmental genomics tools, and we will talk about that it is found the second one is more true. So, the 6 categories of cultivability of microbes are generally observed in the nature. If we take a sample, any kind of sample we take, we found that there are some organisms which are cultured and some organisms which are not cultured. Cultured means they appear readily on an agar plate. If we have a suitable medium, we will find out that some colonies are coming out. And if we take a kind of a cartoon for that, we will see that out of a total community, only maybe 1 out of 100 will be cultured. So, cultures are those organisms that have been successfully isolated and purified in the laboratory. So, these organisms, if we take up this cell or the colony or the strain and try to identify this, you will find that this has been previously identified as a cultivable member most of the time, and these are represented in culture collections. So, we have several international culture collections where these organisms are deposited and they are maintained, and these are often considered, some of them are as a model organism for the basis, and 99% of what is known about the microbiology and environmental microbiology. So, whatever science we know about the scientific basis of our environmental biotechnology, the microbial part of that is basically most of the cases relied on that 99% cases on those few cultivable bacteria, because it is the only in the recent past, like only 10 years or so, we have methods through which the uncultured organisms are also being characterized. So, who are these uncultured, who are not appearing on the agar plates or growing on the agar plates? These organisms could be ones for which no appropriate growth medium has been devised, or possibly the pH and temperature etc. were not appropriate. But this uncultured organism could be cultured provided that we have proper medium or proper nutrients provided, and also the physical and chemical conditions are provided. So, if we are curious, if we are interested, we can culture some of the uncultured members. So, in recent past, we see there is a great effort in culturing many of the so-called uncultured organisms, because once we know that many organisms are uncultured, we are trying to culture more organisms. But so, within the uncultured organism, there could be two categories. One is culturable. Culturable means provided that you have a proper medium and growth condition etc., some of them will grow, but some of them even after that will not grow. Those are the non-culturable, whose physiological state prevents them from being cultured. So, it's kinds of physiological restrictions that these cells or these organisms they have. And within this category, we may have two types. One is dormant. So, like kind of endospores for a bacillus or many actinobacteria, they are dormant. So, their metabolic activities are on a very low level, and they are not willing to grow or respond or reactivate themselves. And the last category could be non-viable. So if we have the entire microbial world in any kind of sample, drinking water, groundwater, lake water, any sample, soil, sediment, anything, we may think of having all these 6 kind of categories. So among these 1, 2, 3, 4 are definitely the viable cells, but the last component could be the non-viable components. So for this part of the lecture, the following references can be used. And in conclusion, some of the major events of development in methods in microbial ecology are highlighted. We have discussed about the great plate count anomaly and the concept of unculturability and the different kinds of culturability, like the 6 different type of organisms, microorganisms which are encountered in any kind of environmental samples are highlighted. Thank you.